In this review, we're going to look at organic molecules and enzymes. So remember, the four organic molecules are carbohydrates, lipids, proteins, and nucleic acids. Carbs and lipids are for energy and structure. Proteins and nucleic acids control reactions. Carbohydrates are sugars that often end in ose. They're made up of monosaccharides, that's the subunit. They provide short-term energy storage, make up cell walls, and some primary examples are glucose, cellulose, and starch. So remember, monosaccharide is the subunit, and that's a single sugar, so for example, glucose. And we can put these together to make more complex sugars like starch, which are polysaccharides. Lipids are fats, oils, and waxes. They're made with fatty acids. They provide long-term energy storage. They make membranes. And that's because they don't mix well with water. And the primary example are phospholipids. So here we have our membrane and we have our phospholipid. Proteins get stuff done in cells. The primary thing that we talk about is they make enzymes, which help control reactions. They're made of amino acids, and these amino acids the chains fold into complex 3D shapes that can work like a tool. They often end in ACE. Some primary examples: hemoglobin, insulin, and remember, ribosomes are made partially of protein. So here's an amino acid. Here's our long chain, which is our protein. It's then going to fold up, and therefore it can work like a tool. Nucleic acids store hereditary information. That's the genetic stuff that's passed down. They code for proteins. They're made of nucleotides, which are made of sugars, a phosphate, and a base. And the arrangement of these bases is what forms the code for the proteins. And again, DNA and RNA are the examples. So here's our nucleotide with its sugar, its phosphate, and its base. And when we put these together and arrange the bases in a certain way, we get a code. Basically, if you know this chart right here, you're good to go. Next, let's look at enzymes. So remember, they're made of proteins. They control reactions in cells, and they do this because they have that folded shape so they can work like a tool. And what they're really doing is they're lowering the activation energy of the reaction, and that helps to speed it up. They get reactions started that wouldn't work otherwise. So remember, we use the analogy of this game. Without an enzyme, it's really hard to win. You have to be really strong. But if you add an enzyme, it's like lowering that bell down, and it makes it easier to win the prize. If we use a graph of activation energy, without an enzyme, there's a lot of activation energy. It's really hard to get this reaction started. With an enzyme, we've lowered the activation energy, and now the reaction can occur. The substrate is what goes into an enzyme reaction. It binds at the active site on the enzyme, and then the enzyme will create products. And remember, we can either break apart a substrate or put substrates together. So here's my enzyme and the substrate. It's going to fit into this active site because of the shape. We're going to add a little bit of energy with ATP, and then it's going to create products. Again, I always like to think about enzymes like tools, especially a wrench. So again, active site, energy, and we create products. Now, some of the characteristics of enzymes: they're specific to a substrate, just like a certain size wrench works on a certain size bolt. They're not used up. You can use a wrench over and over. They need specific conditions of pH and temperature, or else they might become denatured, which means they're destroyed and will never work again. So for example, here my enzyme would work fine because it's got the right shape, but if I have extreme pH or temperature, it can denature, messes up the shape, and now the substrate won't fit. Also, make sure you're able to read these graphs. So if I were to ask you for the optimal pH for enzyme A, all you would have to do, figure out where the greatest reaction rate is, which would be at the peak, draw a line straight down, and a pH of 5 is the optimal pH, or in other words, where enzyme A works the best.